Okay, it's recording, I think. Okay, so I just wanted to go over, sorry, the review. So I already started with a lot of the writing, so the video wasn't that long. Um, but I am gonna pause to give some examples. So first we have linkage groups. So genes that are on the same chromosome, number of linkage groups equals the number of chromosomes for a given species. Um, Chromosomes, another term for a linkage group. Synteny is the physical linkage on the same chromosome. Autosomal linkage is all but sex chromosomes, so just not our X and our Y. Linked genes, genes that are physically linked, they do not independently assort. Recombinant phenotype is a new combination of traits not seen in the parental generation. And remember, this is phenotype. Okay, recombinant offspring have a chromosome involved in crossing over. So that's what gives us our recombinant phenotype. Also genetic recombination is how that happened. Offspring inherit a combination of traits not seen in the parents. Gene locus, the site where a gene is found on a chromosome. Euploidy is a variation in the number of complete sets of chromosomes. This would be like 3N, it would be like a whole new set. Um, Aneuploidy is a variation within a set of chromosomes. So this would be like monosomy or trisomy. Okay. Endopolyploidy is a variation in the number of chromosomes within tissue type. So an example of this is our liver tissues, where liver cells. Okay. Polyploidy is more than two sets of chromosomes. Euploidy is a version of polyploidy. So, and then we're gonna go into viruses, which non-enveloped helical affects plants. A non-enveloped polyhedral infects animals. Enveloped polyhedral affects animals, and then a polyhedral capsid with a helical tail infect bacteria. So that's like the scary guy. It's like I can't draw, but you get what I'm trying to say. That guy. Okay, what are the expected for multi-factor crosses? So a greater proportion of parental phenotype in F2. I didn't really give. Okay, and then be able to state the expected ratios for 2 and 3 factor crosses for keyword unlinked genes. So for a two-factor cross, it's gonna be a 9331. And for a three-factor cross, we would expect would be an equal frequency for each phenotype. That's our null expectation would be equal frequency for each phenotype. Okay, what are the effects of endopolyploidy? So remember, this is our liver cells. And what this essentially does is the more genes you have, the more proteins you have, which is really important for things like our liver, which a lot of our they break down a lot of stuff in your liver, so that's why that's important. What are the common differences between um mitosis and meiosis non-disjunctions? Is it rare or common? What are the differences in phenotype? So it is more common in meiosis. And we would expect for the differences in phenotypes a variation in the number of sets. What is the difference between that? The variation. Oh, this is mosaicism. Um, yeah, okay, mosaicism. And that is the condition in which cells of part of an organism differ genetically than the rest of the organism. Okay, what are the results of meiotic non-disjunction that occur in meiosis 1 and meiosis 2? So, Meiosis one, we have N plus one, N plus one, N minus one, N minus one. So this would lead to trisomy or monosomy. And then in meiosis two, we would have N, N, N plus one, and then N minus one. Okay, what are the effects of tetraploid and aneuploidy in a person with 46 chromosomes? So again, tetraploidy would be for no, 4N. And we have, this is person with 46 chromosomes. So N equals 23. So you would do 4 times 23, and you would get 92. And then in aneuploidy, you would just have N minus 1 or N minus 2. I mean, N plus 1. So you would have 45 or 47. Okay, what are the events that may lead to an extra chromosome? So again, this is crossing over. Well, failed crossing over. This is non-disjunction. And what are the events that may lead to a loss in chromosome that has also failed crossing over and non-disjunction? Okay, what is the null hypothesis for genetic linkage calculations? So the null hypothesis is genes are not linked and they independently sort. Okay, what is a test cross and why do we use it? So a test cross is a heterozygous crossed with an individual that is homozygous recessive. So with homozygous. Okay, and we use it for gene mapping. Use for so based on the test cross, we can see how far or how close genes are based on how much or how little they assort. What does it mean to be an F positive bacteria? So this means that it has the fertility plasmid. Okay, what does it mean to be for a bacteria to be HFR? That means the fertility factor has assimilated into DNA, into the bacterial DNA. So fertility. This means whenever the bacterial DNA um replicates, the plasmid also replicates. What is transduction and how does it occur? So transduction is when a bacteriophage a bacteriophage accidentally packages host DNA, and when it leaves the bacteriophage, it injects the bacterial DNA into another cell. Okay, what is conjugation and how does it occur? So, conjugation is the transfer of genetic material through the sex pilus or direct contact. So, transfer. So, this happens when a protein called relaxosome binds to the origin of transfer in the F factor DNA. It cuts one strand of DNA. The relaxosome then separates the strands of DNA, um, leaving relaxase bound, and then the DNA relaxase complex is pumped out of the donor cell after the transfer of one strand of the F factor. Relaxase then joins the ends of the circle, um, yeah, to produce the plasmid. So remember that. Look over conjugation and how it happens. Okay. Now we're gonna go over what it means to be F prime, F plus, or F positive, and then HFR. So the way I think it's easier to visualize it. So this is F prime, F positive, and then HFR. So in F prime, we have bacterial DNA, right? And then we have a plasmid here. But this plasmid has some bacterial DNA in it because it accidentally picked it up and it kept it with it. And then for F positive, we have bacterial DNA, and it just happens to have the fertility factor. No mixing of the plasmid and the bacterial DNA. And then we have HFR. So this guy, this is the bacterial DNA, right? The plasmid is in there. It's fully integrated into their genome. So what is a heteroduplex repair and what happens? So it repairs the mismatch of one or more bases, of one or more base pair mistakes. So okay, now we have chromosome positions. So we have metacentric, which the centromere is in the middle. Submetacentric, which is slightly off center. And then we have acrocentric, which is more off center. And then we have telecentric, which means the centromere is at the end. And remember, when we have these different positions, it's really telling about the chromosome structure. Okay, now on to chromosome alterations. So for deletions, we have terminal and interstitial. So terminal is obviously going to be at the end. We're just going to have, oops, we're going to take like, probably shouldn't have done. Anyways, we just cut a chunk off. That's gone. We have a deletion at the end. And then interstitial, we have, it's two breaks. So let's say we had a break there. Oh, we had a break there, and we had a break there. So now this whole piece is gone. Okay. Duplication, abortion of the chromosome is repeated due to misalignment during meiosis. So like here, imagine we have A, B, C, and then here we have A, B, and two C's. So that would be a duplication. In inversion, we have pericentric and paracentric. So pericentric is a change in direction of part of the genetic material. And pericentric, it's specifically within the centromere, or the centromere is within the part that switched. So I'll just draw another one to give you like a normal chromosome, and again we have A, B, C, and D. So pericentric involves the centromere. So it would be like A, and then you would have C and B flipped, and then you'd have D. But this involves a centromere, so it would be a pericentric inversion. And then we have paracentric, which doesn't involve the centromere. So it would just be like A, B, and then you'd have D and C mixed. But because it doesn't involve the centromere, it's paracentric. When I think of para, I think of like next to. So maybe that will help between peri and para. Or you could also think like pair the I and peri, it stands for involved centromere or something like that. Okay, translocations. A segment of one homologous chromosome attaches to another. Reciprocal is when there's a two-way transfer. So this would be a reciprocal. So this is when, like this one was originally an all blue one, and this one's supposed to be an all red one, but they kind of swapped. So like that one there, and that one there. So like it was a two-way transfer, so it's a reciprocal. But a translocation can happen when just like, okay, say we have this blue and this red. This red just loses a bit, and it becomes attached. So now this red is gone, but this blue has what was lost. So that's translocation. And now on to the virus life cycle. So attachment, interaction with the cell, entry, um, just enters the cell. Remember, this can be different. It can come through a vesicle or just straight, like, inject itself. Integration. Again, this is not the same for all viruses. Not all viruses do this step. Um, we have the integrase enzyme, which viral genetic information gets into the host chromosomal DNA. So know what that is. In it's one way. So and then we have reverse transcriptase, which is HIV does this because it's an RNA virus, but it has this reverse transcriptase, which can take it from RNA to DNA, and that it's called a retrovirus. Okay, so I mean, synthesis, transcription of genes and translation of proteins. And this is, of course, the virus, the viral genes. Okay, viral assembly, this is um where it's starting to put stuff together. Okay, and then we have the release. This is the lysis of the cell. Which brings me to my next point. We have a temperate phage and a virulent phage. So a temperate phage has both um, latent and lytic cycles, and then a virulent phage only has lytic. So we talked about this a little bit in class, like a latent infection will just kind of stay in your genes and will like only come out like when you're agitated or when you're stressed. So that's what latency is. And lytic is just where it constantly is reproducing and reproducing, and it's never latent. Okay, now we have a three-factor cross. So let's find the middle gene. So to find the middle gene, we're gonna cross. Let me just. So 10 is our double recombinant, and our other are um our parental phenotypes or yeah, genotypes. So So remember, these are our yellows, and these are our blues. And this is how we find the middle gene. So we look for the one that's not the same. Okay, so these are the same. These are both big C's. These are the same, and these are the same. So the one that is our middle gene is going to be A. And now that we know that, we can find our other crosses. And we're going to keep on using the parentals. So again, I'm looking at these. Yes, that's big A. That's a big C. And that's a little A. It's a little C. So this is gonna be so this is our B cross. So knowing that this is our C cross, and by looking at our answer choices, we know A is the middle gene. Use a different color. A is the middle gene, and we know which one is our B and our C cross. So looking at our answer choices, that's the right order of the genes. Okay, now we're gonna find our map units. So if you can remember, our map units from our B crossover are 205. And you always do your double recombinants. I forgot to tell you the total. 1780. Okay, that's 024. And this is our B cross. Now we're gonna do our C cross, which if you look up here, our C cross is 75. So again, A is our middle gene, and our B crossover was 0.24, and our C crossover was 0.09. So This is gonna be your answer. Okay. M, C, and A are all found on the same chromosome. The distance between genes M and C is 21 map units. The distance between gene C and A is 17 map units. If gene A is the middle gene, what is the distance between M and A? So we know A is our middle gene. And I think they give us the distance between A and something. A and C, 17. And if we know A is in the middle, then M is going to be on the other side. And from M to C is 21. So 21 minus 17 is 4. Okay, if gene transfer occurs in a clockwise manner and the origin of transfer sits to the right of pro-AB, which is highlighted here, to the right, so it's a little after 5, how long would it take for pole A to be synthesized? So pole A sits right after 85, and we're around the 5. So if we start at 5 and go all the way to 85, the answer would be 80 minutes. Because from the origin of transfer, it would be 85. But because we got a little bit of a head start here at 6 minutes, um, it's gonna be 80. So yeah, if you have any questions, feel free to email me. Good luck studying on your exam.